So my name is Lauren, and I did a literature review on the ubiquity of the neurotoxic amino acid, beta methyl amino L-alanine, or BMAA for short, around the globe. And for those of you who may not know what ubiquity means, ubiquity refers to the state or capacity of being everywhere, especially simultaneously. So a little bit about me is I'm currently a 12th grade student from Chicago, and I'm interested in biology, neuroscience, and environmental science. And about my mentor, Tisha Luer, she is currently a PhD candidate in biochemistry at the University of British Columbia, and she has done many projects, and I had a variety of subjects to choose from her. And one of her thesis projects was the method development for detection of BMAA in food and environmental samples. And my reason for choosing this topic was I was initially interested in BMAA's link to neurodegeneration, and I wanted to understand and illustrate how widespread BMAA was and bring attention to various potential sources of exposure to BMAA around the world that could increase the risk of neurodegenerative diseases. So about BMAA, it is a non-protein amino acid, and that basically means it's not one of the normal 20 amino acids that make up the human proteome, and it is produced by cyanobacteria, which is arguably the most widely distributed and ancient organisms on Earth. BMAA is produced as part of the cyanobacteria metabolism, but not used to end the cyanobacteria proteome. BMAA was first implicated as a possible causative agent of neurodegenerative diseases like Alzheimer's and Parkinson's disease, and was first linked to a high incidence of ALS-PDC among the indigenous Chamorro population of Guam. It was claimed to have been caused by ingestion of BMAA containing food. BMAA was first isolated from cycad seeds sent from Guam and can be found as a free amino acid or protein bound, which basically means it's already attached to an already made protein. Or it can be misincorporated into proteins, which can lead into protein misfolding, aggregation, and possible cell death. And the protein misfolding and aggregation is a key feature in neurodegenerative diseases. So it's claimed that BMAA can biomagnify through the trophic levels, and biomagnification refers to an increase in the concentration of a substance as you move up the food chain. For example, flying foxes, which can be seen on the right, would feed on cycad seeds, which can be seen on the left, that contain BMAA, delivering a large dose of BMAA to the Chamorro people who ate them as a delicacy, such as making flying fox soup. There have been numerous reports of BMAA accumulated through trophic levels in various ecosystems and food webs. So what I did for my literature review was I did detailed searches through the online database of PubMed, and searches were conducted in English with the following key terms of BMAA and its full-length name. I looked through 566 publications about BMAA and selected publications that detected the presence of BMAA in various sources. I then organized all the citations into tables with the following categories: animals, supplements, and natural health food products, human tissue, water sources and deserts, and plants, and then discuss the implications of the overall collected data. And I'll show you an example of one of my tables, and this is a table about the presence of BMAA in human tissue. It shows where BMAA was found, such as ALS patients in Guam or ALS patients in Canada, and the BMAA type, free amino acid or protein bound, and the different quantification methods, such as high performance liquid chromatography, fluorescent detection, or mass spectrometry. This is a summary of the total number of citations I found in regards to the presence of BMAA in animals. And BMAA was detected in a variety of species of animals, from mollusks to crustaceans and dolphins. And on the right side is a table that summarizes the data of all the different categories. So the data that I collected has led me to believe that people around the world are at risk of exposure to BMAA, where various possible bioaccumulation and transfer routes have been detected, which can increase the risk of human and animal exposure. And with climate change and increase of cyanobacterial blooms, this could lead to a possible greater production of BMAA. And with all the supplements and natural health products that I found, publications that I found, they're not being screened properly, which can also increase the risk of exposure. There has been a lot of controversy and conflicting data surrounding this topic, as there is a lack of validated quantification methods. Not everybody is using the same quantification methods, and some may not accurately quantify BMAA. There was even a critical review that questioned whether this BMAA hypothesis was false or even true. And but with my research, there has been an overwhelming amount of evidence of BMAA being detected. However, there should be more research to accurately quantify BMAA in sources like tissue to ensure that health authorities and the public are provided with the correct information to minimize the risk of environmental BMAA exposure. And my ending remarks is that my mentor was amiable and very helpful, and I'm really grateful to her. Her knowledge and guidance was fundamental to the success of my project. She taught me how to write the lit review because I had never written one before, and she introduced me to different softwares to organize and cite publications. And this experience was overall enlightening and memorable. And that's the end of my presentation. And I just wanted to say thank you again to my mentor, the Talaria Summer Institute, for allowing this to happen, and everyone who listened in my presentation today.